Okay, I think I'm going to start. So my name is Alicia Kahn. Welcome to the NannoTalks. I am happy to introduce a very good friend of mine, Dr. Andrew Bowman. Andy went to school at the University of Nebraska, worked under David Watkins, and then he came and worked for Unocal, and then Chevron, and then Statoil. And right now he is independent contractor, and he will be giving us a talk on the taxonomic revision of the Heliolithaceae. And I'm very excited to see this talk because they're very close to my heart. And I'm also very pleased to see Andy, because even though, we live in the same town, the pandemic has made it very difficult for anyone to see anyone. So it's nice to see you on camera, Andy, and welcome to, it's a nice big group today, so I'm very pleased to see everybody, and welcome. And we're not sure if Andy's Zoom is going to end after 45 minutes, so we're going to keep his talk at about 40 minutes and then give time for 5 minutes for questions. I will pick up the chat, so feel free to use the chat. But again, Andy, you don't need to be looking at that. I'll be taking care of that. And should we actually have the extra 15 minutes that we're not anticipating, we can stay on and continue the questioning. So without further ado, Andy, take it away.
Thanks, Alicia. Thanks for the introduction, and thanks everyone for having me. I'm thinking I'll go through this faster than I initially thought, because I actually logged in a bit before 10. So if there's 45 minutes, I better get going. So here we go. So the title is the taxonomic revision of the helioliths, and the outline of the presentation, just a brief introduction, getting into the methods about sample prep, slide preparation, the analysis as far as routine analysis and mobile mounting, the use of the optical properties that we established, or that we used from previous literature, and then finally the results, the taxonomic groupings, the new and existing genera and species, and then any sort of implications. So the causes of taxonomic confusion within the helioliths. First, the first issue is that many of the species have holotypes that are SEM images, and it's difficult to, often difficult to relate the SEM image to the light microscope image. And an example of this is Bomolithus elegans, and what we're calling a new genus Casedo conicus. And so elegans obviously here has two discs and a column. Conicus has one disc and a column. So that's the separation there. The second issue is that numerous holotypes and paratypes that document species are not compatible. So they're of different species. And also there's been just a lot of misidentified species in the publications as well. And a good example of this is Bomolithus elegans. Let me, can you see my cursor?
Yes.
Okay. So on the two photos on the bottom right, it's not Bomolithus elegans. You can see it has one disc and a parallel-sided high column. That's what we're calling Casedoe. In this case, it's C. pattersoni. And then actually the photo next to that is Bomolithus elegans, because it has two discs and a column. The median disc is wedge-shaped and thin, but you can see clearly there that it has two discs. The third issue is related to the holotypes that exist are often incompletely described or not described sufficiently. And a lot of that has to do with kind of the morphology. So what we're calling Heliotrochus, the former Heliolithus kleinpelli, the discs are very thin and the column is thin. So it's two discs and a column, very compressed. And so you don't get the distribution of mass to allow it to settle in side view that often. So what we see is, we typically see just the top views of the really compressed forms. So as a result, it the past work has been a bit muddled, and of course it's totally understandable considering how these species often settle. So what you see on the right here, these photographs, you have the side view of Heliotrochus kleinpelei on the left, and then these two on the bottom. That's the holotype of C. robusta or Ericksonia robusta of Bramley and Sullivan. And then what we have here is a new genus called Tanromania. In this case, it's T. tunisiae. And this is Tanromania's the form on the right. They have two equally sized discs in a column that is also the same size as the discs. So the same thickness and same width. So it's like a 3-cycle form. And the holotype of E. robusta is, you can see that on the left there, the disc is dim. It's not bright like the Tanromania on the right. And it looks like it has three, on side view, I guess, cycles, but they're not equal thickness. And so they're not the same. And so we'll get more into that here shortly. So secondly, there's biostratigraphic issues with the helioliths. The first is the incorrect taxonomic concepts produce inconsistent and confusing results. So because a lot of these species look very similar or identical in top view, it just causes a lot of problems. So it's really important that we do the mobile mounting, and so that's what we applied in this study. We did the mobile mounting technique on several sections and mapped the structures of all the heliolith species. Secondly, the published zonations lack a lot of resolution, specifically upper NP7 and then NP8, NP9 with the base of Discoestra multiradiatus. And also these applied to the industry, which a lot of us here have experience with. It's difficult to use bases and first occurrences in our work because of downhole contamination from cavings from upholes. So that makes the base of Heliolithus rhytuli difficult to use, and that's a major marker right in the middle of a main reservoir package in the Gulf of Mexico. So we get around this by we have established several new heliolith species that give us a lot of a lot of them are short ranging, and there's a lot of control then in NP8, NP9, and then also NP7. And then finally, preservation can be an issue, specifically where a lot of us have worked in the Gulf of Mexico. The poor preservation of samples makes identification a little bit problematic. Specifically, whether there's a median disc, a thin median disc present, or if it's just a single disc form. And I'll get into that here shortly. So again, using the mobile mounting technique is really the way to go. So these are some examples of the 5 genera that we're using. So 2 of them are new genera. So Casedoes in the top upper left, those are single disc form with a column. And then the other new genus is Tanromania on the bottom. And those are 2 discs in a column, all equal size. And then we've brought in Heliotrochus published by Aubrey in 2014. And so Kleinpelei and 2 other species, 2 new species go into that genus. And then we're using Bomolithus and Heliolithus as well. Those are unchanged, but we've reassigned some species. And so you can see that those kind of magenta boxes show the top view of these helioliths. And you can see that there's some variation, but in some forms, they seem totally identical. So you have to really, to be confident, you really have to rotate these things. So I'll show some examples here of what I was just mentioning. The first example is misunderstood heliolith taxonomy. And so what are these things? Are these the same species, or are these different? They look pretty similar, but they do look different. So the species on the left is actually Heliotrochus kleinpelei. You can see inside view it's very thin. It has two discs. One is the median disc is narrower than the proximal disc. Proximal disc is down here. And then the column is also compressed. Whereas the species on the right is a new species, Heliolithaceae colobos. And this is one disc and a column. So it's structurally totally different. And so that's really the trick, you know, as we go through these. So you have one disc in a column form that looks very similar to two discs in a column form. And the birefringence is the same with both of those as well. Here's another example. This is more, I think, in line. So these look really similar. Once you know what you're looking for, you can kind of separate them. But you can see it inside view then. The species on the left is Bomolithus elegans. It has two discs in a high column, parallel-sided. You see that median disc is wedge-shaped. And then, in contrast, the species on the right is new genus and species Cassedoe micheliorum, and that has one disc and a column. And the birefringence is different in this genus. So in plan view, the column is birefringent and the disc is non-birefringent. And then side view, both the disc and column are birefringent. So out of the five, this Cassedoes are the genus that that doesn't have complete birefringence in both plan view and side view. So it's a combination of number of discs and birefringence, basically. Third example here is about the preservation. So you can see on the left, we have Heliolithaceae rhytuli, one disc and a column flaring. And then on the right, Bomolithus universus, two discs and a flaring column. So, I mean, they're similar size. They both have, you know, full birefringence in top view and side view. And the difference is Heliolithus is one disc, Bomolithus is two discs. So in really, you know, crappy material, which a lot of us work on if we work in the Gulf of Mexico in the Paleocene, it's difficult to differentiate that median disc. And so you could really confuse Bomolithus universus with Heliolithaceae rhytuli if you don't have really good specimens, and unfortunately they overlap in range, so that won't help you really too much either. So just things like this you have to be careful of. I mean, and really nice material, like what we worked in on the paper, it was all ODP and DSTP material, so it was you know, pretty nice. But in certain basins, it'll be a bit problematic. So getting into the methods, this is a map showing the sections in North Atlantic, South Atlantic, Pacific, Indian Ocean, and these were all included in the paper, in the study. And a lot of text here, but I'll just kind of go through it quickly. This is kind of methods about the preparing the slides and the mobile mounts. So the smear slides are just making however you prefer to make your smear slide. You know, that's the method you should probably follow. I'm not sure. It does obviously vary, but you just want to be careful on that. And so you're going to prepare two sets of smear slides, one with cover slips and one without cover slips. And then the one with a cover slip, you can do routine quantitative analysis. And then on the other set, you'll make the mobile mounts. So the mobile mounts will be just the dried sediment on the cover slip without, or the slide without the cover slip. You scrape that sediment onto a new slide, add this really lovely immersion oil that's really extra viscous, and then make it like a slurry with a toothpick, cover it with a cover slip, and then you have to wait because things are going to be rotating really quickly. So usually let it stay overnight. And then when you're looking at it under the microscope, you just tap down on the cover slip with a toothpick or a pen or something, and then it gets the things rotating. And now this is the good stuff here, this image on the right. That's what you're going to want to use. It has a viscosity of 21,000. Not sure what that means, but it seems like a lot, and it's really quite thick. So that's what you should be using, in my opinion. Okay, so we have a couple videos of mobile mounting. One is Casedoe conicus, one is Bomolithus elegans, and they're each about, I don't know, maybe 3 minutes. I thought maybe we'd show one and then we could see both, but I'm not sure. Maybe we can just go one by one. Anyone have a vote? Why don't we go here? These were produced by Osman Verrall. He's posted these on YouTube, and I put some links on the bottom of this slide where there's other species that are also mobile mounted in video and put on YouTube. And there's more than this. There's quite a few. So thanks, Osman, for that. So it takes a while to get going, but so that's plan view, obviously. Can everyone see that okay?
I don't think it's come up yet, Andy.
Oh, really? Okay, hold on a second. Hold on a second. How about now?
I cannot see it. If anyone can see it, write it in the chat.
Oh, you can't see it?
I can see your slide, but not your video.
Okay.
Andrew, I think you need to stop sharing your presentation and start sharing the YouTube channel. So
that's complicated.
Yeah.
Do you think I know how to do that?
Or if you could share your full screen, Andy, maybe instead of just.
I moved the screen and it didn't show up. Yes, I changed screens again. Let me see.
If you're sharing just your presentation, then the other screen with the video will not show up. You need to make sure that you're sharing your entire screen, or
I can't really hear you, Alicia.
Oh, sorry. You need to make sure that you're sharing your full screen, or else you need to stop sharing and start resharing with the YouTube screen.
Okay. Well, why don't we move on? This is going to take some time because I'm kind of screwed up here.
I'll try to pull up Andy and see if I can share my screen at the end.
Sorry, I didn't hear you.
I'll try to pull it up while you're doing the rest of
I see. Okay. Oh, yeah, great, thanks. Yeah, because I think we're going to be a little bit short on time. So, but anyway, there's the links here. You can, Alicia, if you can figure that out, that would be great. But you do have the links here to watch it at your leisure. And I'll just move on. Sorry about that. Okay, so we'll get into the optical properties and the methods, and as far as that goes, we've followed Romain 1979 as far as the methodology that he followed and established. And so basically the extinction lines, the way they curve, whether they curve to the left or to the right, gives information about the crystal configuration and under cross-polarized light. So if they curve to the left, it's been established that that's proximal. If they curve to the right, it's distal side. And then in relation to that, you can look at the colors, the blue and yellow coloring in the various sectors in plan view of the species, and document the color patterns. And that gives you using a gypsum plate, and that gives you information about crystal orientation, arrangement of the crystals, and then you can decipher whether it's proximal versus distal view that way. And then finally, the kind of combination of the two. On the bottom left here, you see the blue is oriented using the gypsum plate. The blue coloring is aligned vertically, and then the yellow color is along the horizontal axis, and that represents, and then the extinction lines are curved to the left, so that represents the proximal side. Again, and then the distal side is just the opposite. And then that's well illustrated by Marzarella and Raffi 2018 here on the bottom right. Very good illustration. Okay, so we verified these optical properties in circular and subcircular non-helioliths, so basically placholiths. And so we just wanted to just test this on our own. And if you can see the highlights that I made here. So you can see the alignment of the blue and the yellow axes. I just made brief highlights here, but yeah, you can see that it works. You see the left curving extinction lines in the proximal view. Here I'm looking at the left with Marcalius and Tauius. And then in the center, you have the right curving extinction lines, and then the blue and the yellow are aligned the appropriate way as well. So we tested it on the placoliths, and then based on that, we were able to have confidence that the helioliths behave the same way. So it's across the entire species and generic level. This relationship does hold up within helioliths as well. Okay, we'll get into the results. So we're using 5 genera to represent the helioliths. So the Tanromania and Caesadoes, so that's down here at the bottom, Caesadoes in blue font there, and then Tanromania is on the bottom right. And Heliotrochus, we've implemented from Aubrey 2014, and then Bomolithus, Heliolithus are the same, are unchanged. But you can see again, the top views are pretty similar, but it's the side views that really show what these things are. So Bomolithus, 2 discs and a high column. The discs are either equal or unequal in width, and the column is variable, and it has full birefringence. So that means the discs and column are birefringent in plan view and in side view. Heliotrochus, 2 discs and a very low column. They all have similar height, and making this specimens appear compressed, very thin. And again, they have full birefringence too in plan view and side view. You have Heliolithus, which is 1 disc in a column with full birefringence, plan view and side view. Caesadoes has 1 disc in a column. The column varies in height and width, and it has partial birefringence, meaning in plan view, the disc is non-birefringent, the column is birefringent, and then in side view, the disc and column are both birefringent. And finally, Tanromanias, those are 2 discs in a low column, all of which have equal height and width. There's a wide featureless central opening, and again, they're fully birefringent as well. And we'll get into some photographs here shortly. So this slide shows the workflow used for first kind of deciphering what group these forms belong to, or these specimens belong to. So we use structure, again, which is the number of discs, 1 disc versus 2 discs. The characteristics of the column, whether it's low versus high, parallel-sided, or flaring. And then the relationship between the discs and the column as well. The size relationships between proximal and median disc, relationships with the column, things like that. And also birefringence. So 4 of the 5 genera are birefringent in plan view and side view. And those are Bomolithus, Heliotrochus, Tanromania, and Heliolithus. And then the Caesadoes are the outlier. They're as I mentioned, non-birefringent disc in plan view, and then everything else is birefringent in plan view and side view. So getting into the Caesadoes, have some illustrations there showing in the photograph showing the curving extinction lines, and then the alignment of the blue sector along the horizontal and vertical axis. Vertical axis for proximal view, horizontal axis for distal view. And we'll go through, I don't want to get too deeply into this, but I'll just go through the primary species and just kind of highlight what the morphologies are of these. So Caesadoe atlanticus, new genus, new species. It's the only elliptical form of Caesadoes. Has a wide parallel-sided column. Of course, 1 disc and 1 column make up all the species here in this genera. Casetoe conicus, which has been reassigned from Bomolithus. You can see it has one disc in the column, so it's not a Bomolithus. It has a tapering column, and it's a pretty good marker. Casetoe grandis, it has a very large form, very wide, flaring, a column that flares distally, and it's similar to Megastypus, but this possesses a gigantic column, whereas Megastypus is a kind of a shorter column. New species, Casetoe camina desneratum. Not sure if I even pronounced that right, but there you go. Has a column that is parallel-sided to slightly tapering, and a fairly wide column that resembles a fairy chimney, which it took me a while to even know what that is. Osman named this one, but yeah, so the column represents some sort of chimney form. Casetoe megastypus has a wide and tall column, but not as tall as grandis. And Casetoe mitchellorium has, again, one disc in a column, as they all do, a parallel-sided column that is wider than it is higher or taller. And that's going to separate it from this species here, Casetoe pattersoni. It has the column height, again, parallel-sided column, tall column, but the height is greater than the width. And that's the key diagnostic for that. And then finally, Casetoe yapesi. This basically has one disc in a column. The column is so low, it's truncated to where it looks like a hand fan inside view. So there's very little free length, if any, in the column. It doesn't raise above the height of the disc. Okay, getting into the Heliolithus. Again, a single disc form. Rhytuli here shown in the illustration. That's really, I mean, it's the type species for the genus, but it's really totally different than a lot of these other Heliolithus as far as morphology. These other Heliolithus have one disc in a column, but they don't have the flaring nature that Rhytuli has. So that's a whole nother can of worms, but I'll go through some of these species here. Heliolithus atkici, it's previously published. That was remained as it is. It's a form that has the one disc in a column. The column is about the same width as the disc. It appears kind of like a pill bug or something. Heliolithus amphibolos, it's a unique form, one disc and a column with a relatively wide central opening. It's a strange and delicate form. The column is indistinct or very low, but you can see that the disc is wider than that column-like structure. Heliolithus buckrii, that's unchanged. It's a good marker and good event. Parallel-sided column, relatively narrow. Oh, I'm sorry, my YouTube was still going, and now there's a commercial. Okay. A parallel-sided column, relatively wide disc. The height of the column varies, and the column appears hollow. It's a wide central canal, basically, in the column. Heliolithus iptemenitiscos. It's one disc in a column, kind of resembles a flying saucer. The column is relatively wide, almost as wide as the disc, and it's slightly tapering, so the species kind of decreases in width distally from disc to column. Heliolithus knoxi, that's unchanged. Again, similar to buckrii, but the central canal is narrower, not as wide as in buckrii. And it's the column can get quite tall or quite large in these species. Heliolithus colobos, it's one disc in a column. The column is narrow and this appears truncated as well. And there's no obvious tube cycle or the central canal is very thin and the tube cycle is indistinct. This has a range, very, very, very short range in upper NP7. It's a really good marker. Heliolithus magnificus has a low column, wide disc. The column doesn't really have much height to it at all, and it appears kind of squished in side view. Heliolithus margarita, another unique form, one disc and some sort of column-like structure in the middle. It resembles a daisy in plan view, if you look on the right here. And this is a great marker in lower NP8, actually. Heliolithus nephroidus, again, has a one disc in a column. The column is almost as wide as the disc, and it has like a kidney shape appearance to it. And then rhytuli on the right, again, it's this flaring nature is not really seen in many forms. In fact, the only other form is in Bomolithus universus, really, this amount of flaring. Again, the difference between this and Bomolithus would be the extra disc. So yeah, rhytuli, it's the results of our biostratigraphy pushed the base or the first occurrence of rhytuli quite a bit older. So that's what we'll talk about here in a few minutes. You have to get through some of these Bomolithus and then a few more. I'll go a little bit faster because all these are just really from the paper that was recently published. So I was kind of going through this really quickly. But Bomolithus have two discs and a column. That second disc is the median disc, proximal disc, median disc, and a column. So Cantabriae, that's reassigned from Heliolithus. It's a Bomolithus. It has two discs and a column. That's right. You can see the median disc is wedge-shaped in side view. The parallel-sided column is pretty wide. It has a pretty wide central canal and tube cycle. The size is variable in these, but the difference between this and elegans is the discs are unequal in width, whereas elegans, the discs are equal width. Bomolithus didymophos, a very strange form, two discs in a column. The discs are about equal diameter, and the column is like rounded, looking like two hills, like dome-shaped. Pretty unique morphology. And that's a young form. That's NP9 form exclusively. Bomolithus elegans. Here you have two discs that are unequal, or sorry, that are equal width, and a parallel-sided column. And it's a smaller form as well. I think it's 5 to 8 or so microns. Again, this is different from Cantabriae in that the discs are equal in width. Bomolithus falklandensis has a very wide column, two discs and a column. The column is very wide, has a wide central canal, and that produces this little opening, moderate-sized opening in the central area. And then finally, Bomolithus universus, reassigned from Heliolithus. We were just talking about rhytuli. This looks a lot like rhytuli, except it has this median disc that you can see inside view, wedge-shaped median disc. Okay, Heliotrochus, there's only a couple species here. Clientelae, of course, that's unchanged. We used Heliotrochus, again, from Aubrey 2014, to represent forms with two discs and a column, all birefringent in plan view and side view. And the discs may or may not be close to equal. In Clientelae, they're not equal. The proximal disc is quite a bit wider than the median disc, but they're all the same height. And so it has this compressed appearance to it in side view. And so that's part of the reason why you see these only in, well, mainly in plan view, is that they're just the distribution of mass doesn't allow, side views to be observed. Heliolithaceae tipera, it's a form with 2 discs and a column. The discs are equal diameter, and again, everything is compressed. And it has a moderate sized central opening. And finally, Heliotrochus starobouti, 2 discs in a column that are compressed. The median disc is almost as wide as the proximal disc, but not quite. So the width between the discs is close to being equal, whereas in Kleinpelli, the median disc is much narrower than the proximal disc. Okay, Tanromanias, my favorite. These are 2 discs in a column, again, very compressed, all equal thickness and equal width. So you can see the side view on the right here, 3 distinct cycles there. So these are all new species. Tanromania billini, it's any species of Tanromania that's less than 8 microns in diameter. They all have wide central openings as well, and the size varies a bit between the forms. Tanromania davidi, this is a species of Tanromania that has a central opening that is 2 times wider than the width of the discs in column. So again, a compressed form, wide central opening. Tanromania deflandrii, it's one of 2 elliptical forms within the genus. So that's a diagnostic feature, that it's elliptical, wide central opening, and it's just a form that has a central area that's wider than the width of the discs in column. Tanromania denisiae, this is any species of Tanromania that's greater than 11 microns. And it has a very wide central opening to it too. And you can see the side view is really distinct too. Tanromania fatme, species with a central opening that's less than 2 times the width of the discs. And then finally, Tanromania noli, it's another elliptical form, and it's very large, greater than 11 microns, but it's elliptical. And the central opening is just very slightly wider than the width of the discs. So that differentiates it from Denisiae, is that it's elliptical and a narrower central opening. Okay, implication of these results. So the results have implications on timescales and zonations, as well as a bit about improved biostrat in the industry, although I can't really show anything on that, but I have some comments. So this slide shows the geologic timescale 2020, the events that are published in that publication. And on the right are events that I added into Timescale Creator from our paper. So you can see Rhytoli is circled on the left, and the GTS 2020. The base is way older than that. We had the base down at, which is the base of NP8, top of NP7, down at about 58.5 million years, which makes it about 1 million years older. And those ages were based on calibration with age depth plots. And so we used the published ages from GTS 2020. A lot of those are bases to estimate ages, numeric ages for these new events. So it's potentially could move up a little bit, but regardless, it's much older, and that makes everything else obviously older as well that relate to that. So just I think more work needs to be done here, and to sort out some of these ages, but there'll be some changes, I think, and I think it's due time for those changes. Ericsonia robusta, circled in purple here on the left. Again, we talked about that a bit. It's a really problematic species. It's often just identified in various ways, and it's not consistently identified properly. And that relates, we're not sure what the age of that is, but it looks similar in age to our Tonromania denisiae, which I've highlighted in the right-hand chart. I think it's in purple. I can't see because now I can see. Yeah, it's over here at about 57.2 or so million years in lower end. And then Kleinpelli top in the left, it's way too old. It should be just below the base of multiradiatus. So I'm not sure if that's some sort of mistake, but yeah, we have Kleinpelli way up here, just above Rhytali, which is near the top of NP8. The base of Rhytali quite a bit lower. And then we have all the species on the right here are new species or reassigned species. So we have much more age control at the NP8, NP9 boundary, which is a good thing because the base of multiradiatus is a terrible marker. And then also NP7 to probably upper NP6, we have a good assemblage as well. A lot of events going on, a lot of short-ranging species as well. Okay, I don't want to talk about that. So regarding the ages we've assigned to these events, this is an order of events that we have, and the black colored events are just the heliolith-based events, and then the light blue events are the non-helioliths, and a lot of those are markers. So those are what we use when we're creating the age depth plot to establish some numeric ages for the helioliths. So as long as we have, this order of events works really well. I've applied it in the Gulf of Mexico already and had really nice results. So the order is really quite accurate, and it's just a matter of kind of pinning down the numeric ages. I think it's a good first attempt, and be curious to work on this a bit more with kind of getting those ages sorted out. And there's a unique interval that we've discovered in NP8, and it's really quite strange. It's we have the top of the Casados up here in lower NP9, that uppermost pink highlighted box there. The assemblage is dominated by the Tonromanias. They're exclusively NP8, NP9. They don't go older. Casadoes and Bomolithis. And then you have an interval within NP8, upper NP8 into mid NP7, where the Casadoes disappear, and it's just a low diversity of helioliths. And there's a lot of holococcoliths preserved. So, I'm not sure what's going on there. Those Casadoes come back. I'm going downhole here, so I'm not thinking the right way, but into mid NP7, upper NP6, the Casadoes actually, if in a downhole direction, reappear, and I can't tell you really if they're even sporadic in NP8. I mean, we haven't really seen them. So not sure why they're disappearing. And then you have a lot of these holococcoliths hanging around, but it's pretty unique that you have two assemblages, one NP8, one NP9, and one NP7, NP6, that add a lot of resolution in the biostrat. But NP8 is this like no man's land, although there's a couple good events in there. Okay, and then sort of industry-related importance to this. There's nothing I can really show, but the Paleocene in the Gulf of Mexico is really challenging, and that the preservation is really poor. It seems like almost everything is overgrown, and that makes observing some of these taxonomic features, like that median disc in Bomolithis, it makes it quite challenging. So obviously in really nice material, you can you can really push this off, pretty far. But in the Gulf of Mexico or any basin really where the preservation isn't that good, you have to use mobile mounting, and you have to just have a lot of patience, because the decent specimens are there, but you just have to search for quite a long time. Usually, sometimes they pop up. But so the issue with the Gulf of Mexico is what we've seen over the last several years is that the palynology works quite well in the Paleocene, but the nanofossils were always kind of not correlating very well, and just being kind of chaotic. So this was bizarre. It's always been something that's bothered me. Is I don't think it's a dilution of by sand related thing, because there's definitely plenty of sand in the Miocene, and there's nanos kind of throughout those sandy intervals, no problem. So to me, it's an ocean chemistry issue, whether it's diagenetic after burial or going on at the time of deposition. Yeah, and it's partly that, and it's partly that the taxonomic issues with top view versus side view. So nanos used to rule in the Gulf of Mexico, and then I had a problem with this kind of issue in the Paleocene. But recent work that we've done in the Gulf of Mexico using our new established taxonomy, almost all the species were recorded, and they were recorded in accurate biostratigraphic position based on the zonation that we published. And then it produced a more robust correlation in the wells. So it's promising. Again, it's very time consuming to do the mobile mounting, especially when the samples are close to barren. You just have to really be patient. But again, we have to be cautious we have to find decent specimens when we're looking at trying to differentiate Rhytali versus Universus, for example. And that's just one example. Okay, I think that's all I have. So we've applied and tested and successfully proven that the mobile mounting technique is working and almost required in certain intervals, specifically the Heliolithaceae of the Paleocene. Again, we've shown that the mobile mounting technique is really the way to go if there's any sort of confusion in identification of top view versus side view. And yeah, that's all I have to say. Thank you.
Thank you, Andy. That was a fabulous talk. Thank you so much. I remember back when I was working with Osman, these many years ago, and he was showing me the rotating technique, and that was exciting.
Yeah.
So it was fun to see that stuff.
Any luck with the YouTube video? I guess not.
I have the video up. I will say, if anybody, and I can do that right now. But if anyone has a question while I pull that up, we could do that concurrently. So does anyone have any questions while I just try to share my screen for Andy? Okay, let's see if I can share.
Hey, Andy, I got a question.
Sure.
How you going? Great talk.
Was it Richard?
I've got it. Yeah, hi. I'll put my camera on. Is that better?
Oh, there you are.
Yeah. Now I got a question about orientation and also about fasciculates. You guys created a new order.
Yeah, right.
The Heliolithaceae.
Yeah.
And you united, you grouped all these guys with the fasciculates based on the presence of a column, right?
Mm-hmm.
And that's what differentiates them from sphenoliths and things like that. I think that was my understanding of what
I didn't get
there.
much into that level, but yeah.
Yeah. So the question I've got, you did all that stuff with placcoliths, demonstrating the orientation using the Romain sort of methods and all that.
Yeah.
and you came to the conclusion that the column is up on these things. So if and you know, I guess the question there is, I mean, these things aren't placoliths. I don't, you know, certainly the way I think of them, they're not using like a VR model. They're not starting from a protococcolith ring like a placolith is. And I guess, you know, if the homology is there, if the column in these things is the same as the column in the fasciculus, does that mean that things like Fasciculus alani, that the point is down?
Yeah, you know,
and that the column is up.
Yeah, no, I really don't know. I've been thinking about that with fasciculus because that's
the question I've got. You know, it seems it.
Yeah. Any ideas from your side on that?
Well, I mean, I guess, you know, it seems hard to believe that something like Fasciculus alani, that the point of the whole thing, you know, they're dart shaped, that the dart faces down. I mean, we're talking about proximal and distal. I think we'd probably agree that these things are some kind of coccolith. They're probably not heterococcoliths. You know, they're not placoliths, but they're probably forming coccospheres. You know, I think that's you know, that's been conjectured by a few people, and I think that's perfectly reasonable. I think, you know, sphenoliths probably do the same thing. We'll never get proof because we'll never find the coccosphere, but
and not with that attitude.
Exactly.
No, but you're right.
It'd be great, fantastic if anyone ever does get one, it'll be amazing. But you know, it's certainly for something like, you know, the dart-shaped fasciculus, it seems that the column would be down, and it seems unlikely that the column would be down in one group, and exactly, and then it would be then reversed in these guys. You know, if it's the same piece of calcite. Sort of, you know, ultrastructurally, if the homology stands up. I mean, you know, if maybe that they're not the same thing, but if they are, then it seems
they sure seem kind of similar, you know. So I was worried about that. I don't know. I don't have an answer, but it needs to be worked on. I think you're doing some work on the fasciculus, right?
Yeah, same sort of. Yeah, I mean, same sort of thing. Except, I guess, I'm looking at the fasciculus a lot more than the helioliths.
Okay.
So sort of trying to link them together, but it's very difficult.
If you guys look at some modern analogs, though, like Gladiolithus flabellatus or Turrilithus latericidioides, and you look at the structure of those coccospheres, because they have similar, you know, robust columns, particularly latericidioides. I think that those give a nice hint for how maybe the nonaconids, maybe the fasciculus, and again the helioliths and the bomoliths, that the structure of their coccospheres. I think that's the kind of thing that we can look at because they are fairly analogous in the modern.
Yeah, no, I hadn't looked at the modern, but that's a good point. Yeah, you did quite a bit of work in that, right? Yeah, interesting. Yeah, it's worth checking, definitely.
So I will share my screen really quick, unless Richard, you have more?
No, I'm good.
Okay, cool.
So I still have my Zoom. It's been over 50 minutes, and so I guess I'm okay.
Yeah, you're a Zoom star. So, so they let you go farther. They let you longer. So here we go. Can you guys see? Can you see this? I have no idea.
If you want to check out some of these videos on YouTube, just search Varel Research. Osmond's posted, I don't know, at least 10 or 15 species in mobile now. So
and Felipe actually has put them into the chat, so you guys can link directly. Can you see what the video that I'm showing?
Do I need to stop sharing?
No, it says I'm sharing, but I guess maybe it's not sharing.
Oh yeah, I stopped. Yeah, you see it? I see it.
You see it? Okay, cool.
Not sure what that blob is right now.
So this
but it's really quite neat. You really understand the structures a lot easier.
And so you're making 2 slides per sample.
Yeah, probably more, because you want to pay attention to if it's too thin or too thick. You know, so you're constantly making other sets because there's something not quite right. But yeah, ideally you'll have a set where you have a cover slip fixed to the slide where you just do the routine analysis, and then the second slide is just no cover slip, and you just scrape that sediment onto a new slide with some immersion oil, and then go crazy.
And presumably, as I recall, like you can't. It really is not a preservable situation, right? You're basically looking at it, and then it's gone.
You gotta toss it, yeah, because they're just really messy with the oil and stuff, you know. Yeah, I don't know what. Okay, yeah, Kleinpelli. I didn't know the Kleinpelli was here. Yeah, you see the proximal disc is wider than the median disc. And then there's some sort of compressed column there as well. But these are hard to get inside view. I mean, this really takes a lot of effort, especially those wide Kleinpelli, you know.
Yeah, this is cool.
Yeah, I think they're each, 3 to 5 minutes, but it's really, really good to see these. And
yeah,
go
ahead, sorry.
No.
Yeah, there we go. There it's rotating. Yeah. So everybody can just go after this talk and look at all of the YouTube videos.
Yeah, that's right. Oh, there's top view. Yeah, okay.
Cool. Well, this is great. Thank you, Andy. I really enjoyed it. I think Richard really enjoyed it. We had a great group today. Does anyone have? I'm trying to find my chat box again so that I can see if anyone sent anything in the chat. Felipe, can you see anything? We have a bunch of Luddites in charge here between me and Andy.
Hey.
I'll speak for myself. I'm sorry. Let's see. So we can get this one out. So, yeah. So if we have no more questions, actually, I see that we are basically at time. So Andy, thank you. This is a pleasure to see your face. And and everybody else who and actually I know sometimes we take a picture, but I don't know how to actually figure out how to do that. So we will sign off. Thank you everybody for showing up, and see you next time. Thank you, Andy.
Yeah, thank you.